Ashwajit Fulzele, Professor and Head Department of Botany, Shri Mathuradas Mohta College of Science, Nagpur. He did his MSc in the year 1993 from Postgraduate Department of Botany. And in the same year, he passed NET examination and he joined Shri Mathuradas Mohta College of Science in 1994. Later, he joined for PhD under the able guidance of Professor R.P. Thakre um, on the topic biodegradation. He is a keen researcher and he has vast teaching experience of almost 29 years in the field of botany, and he is also a regular visitor to the forest, and we have learned so many things in the forest with him with regards to fungi. His field of interest are myxomycetes, leaf litter fungi. Then he has also completed two major research projects, and right now 3 PhD students are working under him, and 3 students have been awarded PhD degree under him. And he is going to talk on fungal diversity, significance, and recent trends in classification. I, on behalf of Rashtrasanta Tukdoji Maharaj Nagpur University and UGC HRDC Center, welcome you, sir, for today's talk, and I request you to deliver the talk on fungal diversity, significance, and recent trends in classification.
Thank you, Dr. Nitin. Good afternoon to all participants. At the outset, I like to express my thanks that to Dr. Dongarwar. Because of him, I will try to present some of the topics related to fungal diversity and the work which is carried out in our laboratory also, and new classification, recent classification. We will try to compare with the some traditional one classification. These are the content. What is the importance of fungi? There is no need to explain much more about this, but we will try to see one or two slide about this. Then regarding the fungal diversity, what is the current status of the fungal diversity that we will try to discuss? Because uh, 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 particularly our area, which we consider as a tropical area, there are many hotspots of diversity, mega diversity hotspots in our area. We are having a huge biodiversity in our country. Therefore, what is the current status of fungal diversity? That if you are able to understand, you will find there are a lot of opportunities to work in this field. Then the before going to the recent classification, we will try to see one traditional classification in very short nutshell, because that is what the most essential thing, particularly who are working in the field, to identify fungi easily on the basis of some handiworks. So traditional classifications are very good for fungal identification. Then what are the new methodologies have been adapted for a recent classification, and due to that methodology, how the fragmentation of traditional groups or the divisions and classes is taken place, and the new type of classification evolved, that will also we will try to discuss in nutshell. And last, I will try to show some of the work which we carried out regarding the fungal diversity in our area. Before entering into the topic of fungal diversity or regarding the kingdom fungi, can you imagine how to understand, how to visualize a kingdom fungi? Because whenever we try to discuss about the fungi, we do not find fungal population, visual fungal population around us, except for some of the big macrofungi we call the basidiomycetes and ascomycetes. Apart from this, there are major group of fungi are microscopic. Therefore, it becomes very difficult to imagine for a common student that how the fungi kingdom look. So, I will show few slides which will help you to imagine how big is a fungi kingdom. This, when you enter inside the forest, you will find that all around our in ambience, huge diversity of fungi present, but they are hidden. So you just try to imagine that these are very big like our plants, and our size as compared to this is very small. So you will able to see that we enter totally into a different arena or totally different area, where apart from a normal animal and fungi, how it looks, just we are miniaturizing ourselves and extrapolating a size of a fungi, then you will able to see that this is the kingdom which is also present surrounding to us, along with plants and animals which we found. These are normally found in the forest, but due to its size and shape, we miss these things. There are some macroscopic and some microscopic fungi surrounding to this. Before entering into this, our today's topic, just try to see what are the these microscopic tiny structures. We call them as a spores, protein bodies, conidia. We are just try to imagine that these things are found surrounding to us. These are the some of the slides which I have taken from the internet. Now, what is the significance of the fungi? There are large number of economic importance of fungi. We know that apart from what the very helpful, beneficial aspects of a fungi, there are many harmful also. But there are only few harmful effects of a fungi. We know that some are very hundreds of species, just hundreds of species are pathogenic to human being, and few are produces mycotoxins and other things. But there are many products which are industrially important. For example, it produces extracellular enzymes, and these enzymes are very important industrially. For example, this lactase, protease, lipase, pectinase, cellulase, renin, all these enzymes which are having in applications, industrial applications, for example, production of ethanol, dairy industry, brewing, food industry, then fruit juice industry, and cheese manufacturing, etc. We know that there are the some secondary metabolites which are medicinally important also. For example, penicillin. That is what the antibiotic penicillin, which is a antibiotic we use, and this only one drug has able to save uh, population of we can call it as a civilization. After the World War, Second World War, the penicillin was product. This fusidic acid, lovastatin, mavastatin, cyclosporin, gibberellic acid. Such many secondary metabolites are there. Due to that, it has immense potential and immense utilization of fungal metabolites. Now, there are many other sources of fungi which are uh, producing a very important industrially and pharmaceutically important chemicals like Taxol, which is used as an anti-cancerous agent successfully to cure the cancer. It produces from a fungi, a large number of fungi. You can see that there is a Taxomyces, tubercularia, Pestalozzi. Many fungi are there. They produce Taxol, which is an anti-cancer product. These all the fungi are isolated or purified from the as a endophytes from the plant sources. So this is plant source are the one of the big source of a new fungal diversity center, and these plants are which are carrying the or harboring the fungi, and these fungi are having an immense potential of production of various secondary metabolites, because we know that the plants are having uh, 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 s- uh, many medicinal content, and these medicinal content which are produced by the plant or the plant source medicinal compounds, we call them, these are actually nothing but the compound produced by the plant with the interaction of these endophytic fungi, and that compound secondary metabolites they produce by a particular plant only after the this interaction with the endophytic fungi. So these endophytes are a big source of secondary metabolites, and medicinally important compounds have been isolated from this. This is the list. The large number of compounds have been isolated. Now we will come to the main topic of our discussion, that is the diversity of a fungi. We know that the diversity of a fungi has been a lot having lot of controversy around it, because there have been claims that there like we know that the David Hawksworth is one of the stalwarts in our family in a community of mycology. He has estimated that there are around 2.2 to 3.8 million species of fungi should be there on this earth. Now, this 2.2 to 3.8, and we can call them as near about the 40 lakhs, 30 lakh, 40 lakh species of fungi should be there. And this was, he predicted this study on the basis of various mathematical and other models and detailed study. We will try to see that in detail today in 2017. That actual range is properly estimated near about 2.2 to 3 point. And we know that current in the current scenario, near about uh, 1 lakh 20,000 species have been accepted, particularly what we call the ICN rules and regulation. According to that, the species which have been named and accepted as a species of fungal species, just 1.20 120,000 species. Now, what they are estimating here, 2.2 to 3.8 million fungi. It is incomprehensible. Worst case scenario, they call this, there are only a 3% of fungi have been named. If you estimate this 2.2 to 3.8 million species if are present here, so only a 0.3% fungi have been named, and 97% fungi are yet to discovered. So recent study which predicted that is what the Lossy and Lennon, that a trillion species of microorganisms are there should be globally. Trillion species, 10 to the power 12. Uh, so 1 trillion is 10 to the power 2. So microbial species, and out of that, they are talking about the microorganisms, and estimated that 1% of this 1% of this 1 trillion 10 to the power 12 species, 1% are fungi. So global estimate of fungal diversity would be 1,000 times higher than the current highest estimate of a 10 million species. If you consider the Lossy and Lennon study based on a computer modeling, they predicted that there are a trillion, 1 trillion species should be there on the. Yes. So this was a paper in which they have predicted that the microbial community, how it will come. There are many types of parameters algorithm which have been developed, and this shows it was a simulation, microbial simulation. On the basis of that, they have predicted that there are 1 trillion microorganisms should be there. While looking to the diversity throughout the world, we found that the diversity increases as we move to different type of ecological conditions. Now, if you see this number of rates total abundance in a human guts also, there are large number of microorganisms. If you move towards the cow rumen, the diversity increases. If you go move towards the ocean, the global ocean, diversity is increased. This line richness abundance scaling relationship it is showing on in this graph. If you move to the earth, on the other parts of the earth apart from the ocean, the diversity increases. So these are the modeling. So these techniques which shows the extrapolation techniques, we call extrapolation techniques, which have been used to estimate the diversity of the fungi. Now, what are the sources? This Hawksworth in 2017 tried to predict that there are near about 40 lakhs species of near about 40 lakhs species of fungi are there, and we just know 1.2 lakh species of fungi, and rest of them are still to discover. So, what are the parameter he used to calculate the diversity? So, these are the some of the point. The publication, then plant and fungus ratio, which similar to plant and insect ratio. On the basis of that, they have calculated that the each plant harbors a fungi. So that is also calculated apart from publication. Then quantitative microecological grid base. We will see further that in detail later on. The method based on environmental sequencing. This is a new field that in the biodiversity, there is no need to culture microorganisms in the laboratory. You can directly take a sample from the environment and put that in the sequencing. You will get the operational taxonomic units, and on the basis of that you can by using bioinformatic tools you can come to conclusion is there any new species is present there or not in the environmental structure, which data which include the plant and fungus ratio and ecological scaling laws. So these are the different methods by which they come to conclusion that there are more than 2.2 million fungi present. So, in the recent classification, generally we found that they have excluded traditional group like a mycetozoa. We commonly called it as a myxomycete group. This myxomycete groups, which we traditionally study in the fungal classification. Apart from this, we know that this Mycota and Hypochytridiomycota, these are also studied by the mycologists only, mycologists and botanists only. They are not a distinct group as we study the traditional classification. But now, in this estimate, where more than 2.2 million fungal species have been predicted, these groups have been separated, where Myxomycota and Oomycota. This Oomycota now we include under the Staminopilla. So what the evidences they have given? So first evidence is from a publication array. So number of new species described in each decade. So this is data given in the Index Fungorum, where you can see in each decade, right from 1750 to 2010, how many species have been added. On the y-axis, you can see the number of species, and right from 1750, you can see that there are every year increase in the deposition or the explanation of new fungi have been added. So 1750 to 1860, this was the ascending phase when the study of naturalist starts and the fungal description, particularly in this era, we know that the naturalists used to study the macro fungi, particularly basidiomycetes, ascomycetes, but they were try to describe different fungi from throughout the world. Then suddenly we found there is a steep phase of a description of a fungi comes after 1870. That was a time we called it as a discovery of or the invention of the microscope. So after the microscope discovery, microfungi added in our account of diversity of a fungi after 1870. So you can see the steep phase, and later on we found that in the 19th century and in the 20th century, it was also a regular phenomena that the new species are continuously adding every years, except for World War II region when the scientific study almost throughout the world it was hampered by the war. But we found that the fungi are added every year uh, by this new fungi. I am talking about the new fungi. The last 40 years, if you look that there are more than around 1,300 fungi, new fungi have been added during the last 40 years. You can see that this is the average. Sometime it may increase, sometime you may see that there is a fall. But from 1975 to 2015, it is average that the new fungi are continuously have been identified and they have been put forth to us. So it is not like that the new fungi, the scientists are working and they are not discovering new fungi. It is a continuous phenomena. This is the number every year this tribe, and this is well calculated graphs have been given on the Index Fungorum website also. So near about 1,800 fungi have been identified. We can call it roughly 2,000 fungi have been identified. Rate have been increased since 2010. Now, this surge in the number of identification of a new fungi, it is largely attributed to increase in a molecular techniques. Particularly, the sequencing techniques have been now commonly used to identify fungi. So, every year, near about 2,000 new fungi have been discovered. So, now if there are large number of fungi and millions of fungi, 2 millions of fungi they are predicting, 3 millions of fungi they are predicting, then why we are describing near about 2,000 species? So, this point you have to remember. 2,000. If there are millions of fungi, then why we are not able to describe lakhs of hundreds and thousands of fungi every years? So that is why this diversity particularly located in the tropical regions, and the collection of the sample and describing new fungi or the traditional work of a fungal diversity is nowadays it is not a very popular work. Therefore, these fungi which are present in the our surrounding in the atmosphere, but due to the less exploratory type of a work is going on, therefore this fungi estimation of fungi, rate of estimation of fungi or identification of fungi is less. What the new tools are available by which we can identify this fungi very fast. Earlier we used to identify fungi only on the basis of a morphological character. Now we are having new tools, that is sequencing, DNA sequence. The, the number of newly named species do not, however, equal to the number that we have been truly described for the first time. Now, this point, one thing we have to remember, this is one of the drawback in the study of the mycology, that many fungi have been published every years, and near about 2,000 fungi are publishing every year. The scientists or the workers give the new names to the fungi and they directly publish as a new species of the fungi. But it shows that there is a lacuna because of non-availability of the sequences in the gene bank. The new workers usually found that if the sequence is not available, that means this fungi is not reported, and they give the new name and publish. So that is one of the drawback, and therefore many of the species which are published, they are already explained by some other workers, but the description is in the literature. Nowadays, on the basis of sequencing, they try to explain these things. So, species concept have been changed recently. In genera where morphologically identical fungi were regarded as a different species if they occurred on a different plant species. We found that the one fungi is present. Suppose Cercospora. Cercospora. So, if the Cercospora is causing disease to the 10 different plants in our locality, generally people give the name 10 different name to that particular fungi as per their host range. So this also creates a confusion. Nowadays, as per the new study, people are try to compare that if they are showing 95% or 99% sequence similarity, then they should be considered as the same genera and same species. It should not be called a new species. So this is what the new technique. Now, what I was talking about, that the analysis of the data which showed the total number of existing species here, if you compare here, the name runs at around 2 to 3 times more than the currently accepted species. What is the meaning of that? Now you can see in this given graph there are these dots. These dots indicates the accepted species of the fungi. Accepted species means they are uh, have been as per the ICN rule of a nomenclature. These fungi have been accepted, but you can see that in the 2010, near about the 50% more fungal new names have been given. So these names are rejected, and on the lower side, up to these names have been accepted. Why these things are happening? Continuously, there is a false data is also deposited by workers on the only on the basis of a few characteristics. They try to call them as a new species, but actually, already that species have been described. The main purpose of that is they are going only through the sequencing pattern, and in the gene bank near about 30 to 35 thousand sequences are available, and we know that about 1,20,000 names have been given to the fungi, so that needs to be rectified. Now, evidence from the species recognition study. So, just now we have studied the on the basis of references, literature, how the diversity is described. Now, here approximately 1 lakh known fungi have been described. 2008 data. It shows that there are 1 lakh species have been identified, and out of that 40,000 are saprophytic, 20,000 are phytopathogenics, mean they are causing diseases to the plants, phytopathogenic. And near about 20,000 species of fungi are out of this 1 lakh species explained in the 2008. They are lichenized, means they show a symbiotic association with the lichens. They are also a very distinct group of a fungi. They do not found normally around us. Then there are about 8,000 species have been identified as a ectomycorrhizal. Then there are 5,000 endophytic, then entomopathogenic. These are the different ecological niche where we found a diversity of a fungi: aquatic, endomycorrhizal, then marine, soil, and human pathogenic. So there are only 100 fungi which causes diseases to the human. We can call them as just 0.01% of the total this. So using this weightage, overall arithmetic means the post priori, that the amount amounted to 11.3, and this mathematical calculation that already accepted fungi may contain up to 10 times as many species as currently recognized, resulting more than 1 million estimate of the species. When they calculated arithmetically by using a different computational model, by studying various ecological niches, they come to conclusion that there are more than 10 times more fungi definitely there. 1 million species should be there in nature at least. Remarkably, this is about 4 times higher than the current estimates synonym ratio of 2.6. The large proportion of these unrecognized species have no name available. So evidence, next evidence which has been given to support that there are more than 2.2 to 3.8 million species is that the study of a plant fungus ratio. The plant fungus ratio, the most most common, mostly from two sources. One is newly discovered species by mean of traditional inventory method is a little studied areas and habitat. That is what a traditional study of a fungal isolation identification from a various on the basis of morphology. And the new study, that is what the newly discovered lineage through environmental sequencing. Environmental sequencing. Initially, I have mentioned about this that without culturing, nowadays it become difficult that many microorganisms are non-culturable. If you try to culture them in laboratory, they do not grow because they require a very specific type of ecological niche. Therefore, a direct sequencing technique, environmental sequencing technique, is been carried out. So these are what the fungal plant ratio is used to identify this. So widely cited and accepted fungal plant ratio that derive global estimate is the 1 point million species of fungi based on the information from several independent sources. So when they try to see that the one plant carries how many fungi as a symbiotic association or in association as a endophyte, so they found that there are more than 230 species have been been carried out by internally by plants. So as a endophytes or in symbiotic association with the plant. So that indicates calculation the 1.5 million species are there. And the fungi reported from all the habitats, that was a study carried out in the United Kingdom by Hawksworth. How many plant species are there, and if the plant species carry the such and such number of species of a fungi, then how many fungal species should be there in the country. So number of host specific fungi on a particular plant species, they calculated the percent of a new species being discovered, the the extrapolation to the global level, and then they extrapolated mathematically and computationally that there are suppose a 400,000 plant species are there, and if you multiplied by the more than 230 species per plant which is associated with the plants, then they found that the 1.5 million figure was considered as a conservative. Conservative. They are not taking the maximum amount. They are taking the minimum numbers. Minimum numbers. The fungi, while number of fungi from all sources, including organisms other than plant and soil, the uh, we found that the such as the millions of insects are there and they are also carries a fungal diversity. So this number automatically become a very huge. So some figure used in this early extrapolation and then subsequently have been revised, and they found out that the vast number of insect fungi had not been considered. Just we are talking about the fungi associated with the plants and other sources. As a consequence, some revised estimates range up to 2.7 to 9.9 millions. You can understand 9.9 million fungal. This is the estimate. This is the estimate they are carrying out. So diversity is a very big, if you compare this to uh, different ecological niches if you try to. Now, the next evidence they try to collect from the environmental sequencing. Just now we have studied about the plant fungi ratio, and also we have added that there are the huge number of insect. They also harbor large number of fungi under it. So next is the evidence which comes for in support to this view that there are near about the uh, 4 million species of fungi, 40 lakh species of fungi, that is from the sequencing of the environmental sequencing. The molecular sequencing data which we have obtained from environmental sample, most commonly from the soil, but also from the water, plant tissue. They have estimated the sequence they obtained, and they try to match the sequences with the data available, sequences available in the gene bank, and they found out that in the gene bank there are just 35,000 sequences have been available. The most comprehensive study of a soil sample to date, this was done by Tederso. They just took the 2 gram of soil sample. It was a big study from a pooled soil, 5 centimeter deep core taken from 365 global sites. 2 gram soil collected from 365 areas throughout the world, and they obtain 80,000 sequences, different type of a fungal OTUs. So this operational taxonomic unit, they use to separate the 98% sequence similarity for the species. So if the 98% 98 and above sequence similarity is found, they call them as the same species. But if the 98% and the below that, they consider them as a different OTUs, operational taxonomic unit. So from just they collected 2 gram soil from 365 global sites, and they got the 80,000 species. That means the if 35,000 species data is available, more than the 40,000 or 40% of the sequences are not named yet, if they consider only the 2 gram soil collected from the 365 areas. So compare with this 120 lakh is formally recognized species. In 25th November 2060, there were only 34,878 named fungal species in the gene bank. So near about 35,000 species have been named in a gene bank, and out of 120. So data is not available. But further, there were 94,000 species level OTUs with no name. So this is a very big work. So this figure have been increased by 68% and a staggering 1,117% respectively in the past 5 years. So if you compare it near the 2005, 2015, this OTUs, which are the what the samples collected from the environment directly, they are the not described yet. So where are these undescribed fungi? Where are these undescribed fungi? If there are at least 1.5 million है ना, करीब करीब अपन उसको बोलेंगे कि 15 लाख स्पीसीज अगर फंजाई की अर्थ के ऊपर है, near about 1.15 millions fungi are there on the known. We have just explained till now 120,000 species. And where can we found this missing 101.38 million species? So near about the 14 lakh species, where are these species? Why they are not described? The three major sources of unrecognized fungal diversity. One particularly tropical regions. Tropical regions. In the initially, I have mentioned this point that these species are mostly restricted to the tropical regions. So the our area, the tropical region of the world, is harboring or carrying the most of the fungal biodiversity in their hotspots. The ecological cryptic fungi that occurs in the environment but usually do not manifest themselves in via discriminable structure. So these fungi which do not show any type of morphological structures or fruiting bodies, but if you take the sampling, environmental sampling, we found a huge diversity is found in this. So this biodiversity hotspots are the major source where the these fungi have been found. So estimated that over 250 species were associated with one tree. Near about 250 species associated with one tree. If there are more than 4 lakh species of plants are there, then you can understand. So 632 macromycete species in Colombia Amazonica forest, 52% could not be identified on the species level. So various authors have studied, they try to confirm this factor, whether there is a huge diversity is found in the forest. So they collected the plant samples from the Amazons, and they found out that while isolating the fungi associated with each plant, the 52% fungi were not identifiable. The other study, they collected 1,430 basidiomes during just 4 collecting expedition in South America. Means basidiomycete species in 4 expedition they carry out in this South America. It also comes under the tropical regions. In the 4 tours, 4 comprehensive tour, they collected near about 1,500 species. So that indicate that the fungi are there either by this environmental sampling or by the morphologically identifiable. So little explore habitat. There are many ecological niche yet to be discovered. For example, there are many bryophytes, many algae, endophytes, fungi which are found inside the vascular plants. Then many tropical foliocolous, then fungicolous, fungi found on the fungi found on the leaves, fungi found on the other things, the mammal gut, insects, then exoskeletons of the insects, then on the rocks. So these are the different ecological niches, deep sea, ocean sediment. They are still not discovered for their discovery. So these cryptic species which are not found, these are generally missing because we suggest that the number of known fungi might rise by the factor of 5. So these are all the evidences which are the existing reference collection. So reference collection, they have identified that the various the 70,000 undiscovered plant species are there. Then suggested that 20,000 fungal species have been collected but yet undescribed. So all these data support that there are the large number of species which are not discovered yet. So all these evidences which we are mentioning, they they support that there are more than 1.5 million species of fungal species, and it was predicted earlier possible. Now they come to conclusion that the estimation may go up to 2.2 to 3.8 millions. That is what the 90%. Then we will come to the classification. Now the here I am finishing the diversity. One point you have to remember that the fungal diversity is a very big area, and the young mycologists need to work for this, because we know that all the facilities mostly for the sequencing and other types of identifying technology is available in the western countries, but the fungi are found in the tropical region. Now, they usually talk during their presentation that this work must be carried out, the workers from the tropical regions. They have to train themselves to study the diversity from the different ecological niche. which are available in the tropical region. So we can able to confirm that whether there are more than 1.5 million or up to 4 million fungi species are present or there. So huge opportunity for the new workers is present. Now, this one classification I am mentioning about what the answer, because it is most important for the fungal identification. Like we use the classification, simple classification like Bentham and Hooker for our plant identification in the field. Though this classification given by the answer is a very comprehensive. He included the Myxomycota in the fungal kingdom as a separate division, and the Eumycota, in which these are the different groups. We know that these divisions I am taking little bit fast, so we can complete and we can spare the time for the matters also. So, in the division Eumycota, where earlier we used to call them as a true fungi, and Myxomycota as a what the plasmodial or pseudoplasmodial, we know that these are the Myxomycetes groups. These are near to the protozoan groups, but traditionally they shows the fruiting bodies like a fungal sporangia. Therefore, no microbiologist, no animal zoologists study these myxomycetes. So this is our traditional responsibility that the myxomycetes was included in our fungal kingdom, and we used to study, and still we study myxomycetes under no other peoples include myxomycota in their syllabus, either zoology, microbiology, but it often found in our syllabus only. And traditionally Eumycota, they divided them into these different sub classes. We know that the Masticomycotina, Zygomycotina, Ascomycotina, Basidio, and Deuteromycotina. These are the traditional group. Now we know that Masticomycotina, in which the oomycetes groups come, and Deuteromycotina, in which the imperfect fungi groups comes. So this is the based on the perfect state spore what produce inside in the life cycle during the after the fertilization. So zygospore, then basidiospore, ascospore, and if no perfect state spore was found, it was convenient to kept in a deuteromycotina, so we can identify this fungi. So this is the classification which was given by the answer that the four different classes have been included in the myxomycetes. I am not going to discuss the distinct characteristic features, but they are very useful, and under a simple microscope or by observing fruiting bodies, you can identify different types of cells and the plasmodia formation and the sporangia formation. You can classify them into cerato myxomycetes, acracio, hydrodictio myxomycetes, and plasmodiophoromyxomycetes. Then this was a oomycota, mastigomycotina, he called, in which the perfect state spore was a oospore. And in this, they includes the chytridiomycetes, hypochytridiomycetes, and oomycetes, depending upon what type of flagellar characters present in that. So it is very easy to identify different fungi, and for particularly in this laboratory study, such type of characters are very useful. Zygospore in zygomycetes, ascospore in ascomycetes. Then on the basis of what type of a fruiting body is present, fruiting body is absent teleomycete, hymenomycete. If the basidium hymenium layer is exposed, gastromyces if the hymenium layer is enclosed in the fruiting body. And the last is the deuteromycotina. So this was a classification. I am not going to discuss about this. This is a very useful. Now in the recent classification, now there are mostly the 18S RNA sequences are studied particularly, but there are now a multi-gene hypothesis have been evolved, and to prepare a perfect classification, you have to carry out the sequencing of many other sequences of RNAs apart from 18S RNA, mitochondrial, then flagellar, mitosis, then during the transcriptional factors, these techniques have been used. So, as a new classification, fungi have been considered as a distinct group, and in that it comes near the animal. And the you can see that at the base, amoebozoa, where the myxomycetes group is present, and the oomycetes have been separated from the kingdom fungi into chromista, where this comes under the hypochytridiomycetes. So, this myxomycetes, Straminopilla chromista, and mycete, such names have been given to the kingdom which earlier all these groups, myxomycetes, oomycetes, and the other groups of the traditional perfect state producing fungi includes in a kingdom fungi. But now these three kingdoms have been prepared, and on the basis of molecular sequences, these classes have been earlier we used to call different orders of the classes. They have been raised to level of uh, division, sometime subclass, division and subclass. For example, chytridiomycete, then blastocladiomycete, Kixeliomycotina, then Mucoromycota, Glomeromycota. So this Glomeromycota, Kixioomycota, Mucoromycota, earlier these are Entomopthoromycota. Earlier we used to include in a Mucoromyces, or Zygomycetes. Now they have been separated on the basis of their dissimilarities in the sequence. They have been raised to the level of this. So this new classification, recent classification, which was proposed by Vijayvardhane, there are about 115 authors have been there, and they use a multi-gene technology, multi-gene tactics for the preparation of this classification. And where they have given a new classification 2020, and a recent modification is also have been, some modifications have been also included. Within 10 minutes I will try to finish. So these are ITS, LUS, then SHU, TF1, these transcriptional factors, ribosome, polymerase. These are what the quite conserved domains for the sequencing, and now this gives the more comprehensive similarities to describe whether they are same species or they are different species. Such technology of sequencing are now we use. So, in the classification, you will find that the nowadays this eukaryota, in which the fungal kingdom comes, archaea and bacteria. These are the two separate domains of a living organism apart from the eukarya. Recently, Geeta Naik, Indian scientist in UC Berkeley, have proposed that she merged the archaea with the eukarya. This classification will come slowly, because recently we consider that the bacteria, archaea, and eukarya, these are the three domains. But she, under a very comprehensive study that Geeta Naik has shown, that the archaea is come here at this level, at the base of the eukarya, and you from the bacteria, through merge of a chloroplast and a mitochondria, plant and animal kingdom or eukarya has been evolved. So this domain has been uh, now we are talking about the eukarya, where our fungi group is present. So animal, this is what the tree, phylogenetic tree, has been developed from the data which is present in the uh, 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 gene bank. Here you can see this is what the fungal kingdom comes. In eukarya, the fungal kingdom. Ophisticonta and Myxomycota will go on the base on this. So they are quite separated from each other. If you enlarge this further, you will find that in the kingdom fungi, this Basidiomycota, Ascomycota, and there are the group which are quite distinct, called as a fungi incerti sedis. That means uncertain positions are in the classification, which include Mucoromycotina and other things near to Mucorota. That is what the Kixiella. glomeromycota, another thing. But there are large number of unclassified fungi. At the base, you can see these are the sequences which have been deposited in the gene bank, which are not having any name, any class. They are still to name. So such data has been depositing continuously in the gene bank. So this is what, if you go further, you will find that in the groups, this is Saccharomycotina and Ascomycetes. These are the new names. These are traditionally we used to call Ascomycetes, and in Ascomycetes we used to call Pyronomycetes, then Plectomycetes, like this. Now, totally new names new terminology have been evolved, like Saccharomycotina, Pegigenomycotina, like this new terms in the Ascomycetes. So in the Ascomycetes also, this on the right hand side, you can see the large number of fungi which are still not described, but gene sequences has been deposited in the gene banks. So this is also, you can say, the all these rounds are suggesting that these are the groups which in each uh, class or subclass or subdivision, there are large number of fungi which are, which the environmental sequencing has been directly done, and the name is not still given. So these fungi need to be isolated, purified, and systematically they have to name. Hmm. Now here, the other group, that is what the in the dicaryota, which is what the environmental sampling here they have been mentioned, and you can see here the some entomycorrhizal fungi. This is the separate group, and this is the Basidiomycetes, in which Agaromycotina has been in Basidiomycota subclass subdivision has been, or subphylum you can call it that. Then the Puccineomycotina. This is the new classification, recent classifications. So you still have Genomycotina. Now you still have Genomycotina, Puccineomycotina. They have been separated. Exobasidiomycetes. So this is a quite comprehensive study has been given, or you can understand that the classification, beautiful classification, can be prepared on the basis of these gene sequences, because the branching is so beautiful that you can separate or arrange different genera, different family, different order in a sequential manners. So earlier the arrangement of the families and the orders were totally depends upon the expertise of the workers or the scientists who were preparing the classification. Now, this gives automatically arrangement, sequential arrangement of the families and order can be given. Here you can see that the Ophistichanta, in which the fungi group was here in this region, and this is the Ophistichanta, and Eukaryota, one branch is coming towards this SAR group on the lower side. On this lower side, you can see that there is a one group SAR, that is a Stramenopilla, and in this Stramenopilla you will find there is a group Oomycota. So this Oomycota is totally quite away from a traditional fungal group, which is what comes above, exactly opposite to that region. So therefore, this is now considered as a separate group, and it has been delimited from the fungal kingdom. They have separated, and they have, and the myxomycete, which comes under the amoebozoa. These are the new names, and here you can see that the Myxogastria is one of the group in the amoebozoa, where the myxomycetes type of a groups comes. That is what the Mycetozoa, Myxogastria. Here we are going downward and downward. Here you can see that the one red line, that is what at the base here, SAR, here we have seen that the Oomycetes group, and here you can see that the Myxogastria, here the Myxomycetes group is coming, and in this Myxogastria you can see that these are the Dictyostelia, these are the groups which traditionally described in the under the different classes, or described by Ainsworth under the different classes in the Myxomycota. So it is one of the group. Myxomycota is one of the group in the Amoebozoa. So it is a protozoan species. It is not a fungi. So it has been totally separated. Now the This is the classification given by the Vijay Vardhane, and then I am just mentioning they have created many divisions, many phylum. Now we use the word as per an ICN, International Code of Nomenclature. ICBN is now abolished, and ICN is used to explain or to describe the nomenclature of plants and fungi both. Earlier, we used to call the International Code of Botanical Nomenclature. Now we called it as ICN. And as per the ICN, the phylum is a valid taxa or ranking. We used to call it as a division earlier. Now you can see that the new names are coming out. These are based on the molecular and molecular data, and they are quite distinct from each other on the basis of similarity. How much sequence similarity is there, they are now considering giving the ranks. If they are showing the 50% similarity and 50% dissimilarity, they raise to the level of classes or the divisions. This new terminology have been comes, and you can see that the earlier there were a few classes. Now in Basidiomycotina they have created a large number of classes. Here large number of classes. In the earlier previous slide you have seen they here also large number of classes have been included. These are the classes in the ascomycete. These are the classes of the basidiomycetes. Then you can see that under this the new phyla or the divisions have been created in the new classification. Blastocladiolomyces, Calcareospora, and Chytridiomycota, Polychytridiomycota, Entomophthalomycota. These are the divisions. Earlier, we used to include in our traditional Mucoromycota, the Entomorhizomycota, Glomeromycota, Kixiomycota. These were the orders earlier. As per the earlier classification, these were the orders. Now they have been raised to a distinct group. So these are also large number of division, and this is the new classification 2020. They have given uh, more than 20 to 30 divisions. Earlier we used to call a one division fungi, and in that the subdivision were myxomycetes and the eumycota, and eumycota few classes were there, and in the subclasses now there are. This classification shows a very uh, drastic changes. Now, a last point while discussing that the what for a new researcher or the who are interested in a mycology or who are specialized in a mycology, what type of a work they can carry out in our area. For example, I will show you the some of the slides which we have carried out work. One of the student, Priyanka, have carried out the work from for only the hypomycetes of Deuteromycetes, Deuteromycotina group from Nagpur district, and we were able to isolate more than 250 species of hypomycete. These are the few slides I am showing that such a beautiful fungal diversity which you might not have seen earlier, but they exist around us. So those who are interested in fungi or specialize in fungi, they just try to explore one or the other groups in detail, and by simple culturing techniques, you can isolate, identify various types of fungi from your surrounding. This fungi you might not have seen such a beautiful diversity. Sometime while preparing culturing, we get cultures uh, uh, of mixed culture, because it become very difficult to isolate fungi from a leaf litter, forest leaf litter. So we directly when we plate on a agar plate, we keep the forest leaf litter, a mixed culture comes. And when we prepare one slide from this one petri dish, I have isolated more than 18 fungi from one slide only. One slide from one petri dish. One so 15, 12 fungi is a very common. So huge diversity is present in our locality. Large number of new species are there. If the molecular sequencing technique or the facility is available, you can publish very easily diversity from our area. Nowadays, paper wants a molecular sequencing technology. You might have seen the Beltrania. I have seen half a dozen species of a Beltrania and near groups of a Beltrania. These are the some of the conidia which found beautiful conidia which were found in these regions. So this is the slide. Then there is another thing that the you can study a particular group. For example, Sheetal has completed her work only on one species, that is what the Chitomium. We were able to identify 22 species of Chitomium from one forest or the forest reserve area from the surrounding uh, National Tiger Reserve project areas. These are the beautiful photographs, and these are the data from the thesis of the Sheetal. This is just a few photographs I am showing you. We have carried out large number of identifying by various methods by morphological study. Then we have carried out some scanning electron microscopy. That facility is also available easily nowadays, and you can prepare identify this accurately. There are many compounds which are produced secondary metabolites. They shows antimicrobial activity. These chitomium species, so we just try to see whether they produce certain types of chemicals or secondary metabolite, and they show antimicrobial quality, and we found that such type of thing. So one species, if you take one species and try to isolate that one species from a particular area, so this is also a very big type of a work. Then endophytic endophytes. These endophytes are what you can see, they produce various alkaloid terpenoids, various types of anti-cancerous drug like a Taxol. So such type of metabolites you can extract from the various types of endophytes. So we carried out a work on neem uh, plant, because neem is a extremely we well known for its medicinal property. So we try to isolate the fungi which are associated internally associated with the names from the bark, from the leaf, from the petiole, from the stem, various parts of the plants. After taking a well precaution that the external flora should not be there, it should come from the internal sources. And we were able to identify more than 15 different types of species, and there are a large number of unidentified species for which we for at least 5 species we have carried out sequencing, and on the basis of sequencing we were able to identify some of the new species from that, and then we try to extract some secondary metabolites from that fungi, and that secondary metabolites from these are all the endophytes which isolated from the neem plant, and they have been identified on the basis of some of the morphological basis and some of them on the molecular basis sequencing we have carried out. And we found that they've produced one very important drug, lovastatin, that is what hypocholesteremic drug, which is having a highest demand in the market. And this drug was also we after isolating this fungi, we try to identify whether these fungi are producing capable to produce lovastatin or not. So we found the various concentration of lovastatin is by various fungi. Some of the fungi show a huge quantity of lovastatin production. HPLC analysis was carried out. You can see that the lovastatin, this is the peak of lovastatin when we compare it with the standard lovastatin. So such one chemical from a one plant, we isolated many fungi, and we just try to see whether it is able to produce lovastatin. For example, Taxol, anti-cancerous drug. Now this is a hypochlorous. Each heart patient is recommended lovastatin or statin drugs. It is essential for this and having a huge market value, billions and billions of dollars. So such type of works can be carried out by our researchers, which are mm, found in our locality, which is a tropical area. Now it is our responsibility that the such type of a work should be carried out. I like to stop here because already more than 1.5 hour is already we have completed. So if any question you can ask, and I hand over mic to the Dr. Dongarwar and person.
Participants, if you have any query, please ask.
Thank you, madam. Hello.
Any question?
Ah,
Dr. Fulzele, sir.
yes,
Can you, I am audible?
yes, yes, yes,
Uh, sir, uh, myself, Dr. Rakesh Thakre. And uh, I would like to congratulate for nice presentation on the
Thank you.
classification of fungus. And I want to share something.
Yes,
Uh,
yes,
like you, I was fortunate to have the guidance of Dr. Thakre from your department, who
yes,
was
yes,
your
yes.
guide
He
even.
was my guide.
Yeah, he was. I don't know whether he's
He
there or
is
not.
very
He
well,
has
and he is in the Nagpur.
right, right. So he is the best mycologist I have
Yes,
ever met. My guide, Dr. Vadhir sir, advised me to go there because Vadhir sir was the virologist, microbiologist basically, and
yes,
I was having the topic on fungal production of laches from basidomycetes, right? So I
yes,
was nil at that time, uh, as I'm the student of microbiology. So I know what is bacteria, what is virus, and all. But I was very afraid of fungal species and all. So Dr. Thakre, um, you know,
yes. He is very helpful,
he
and
was very much helpful, and he makes me to love fungi. And after that, I did lots of work on fungi. I have isolated 365 different basidomycetes from,
yes,
you know, wooden rods,
substrates.
white rot
Wooden
causing,
substrate.
yes, yeah, wooden
Decomposing
substrate,
wooden.
soil, white rot causing fungus and all. So, but out of that 365, I got three species having a higher production of laches.
Yes.
Okay, so for that purpose, I need to classify that fungus on the basis of the molecular characterization.
Identification.
So I went to 18S16, 18S RRNA technique, and I got a 99% similarity
Hmm.
with existing what we can say existing with existing culture.
Ah,
But if some species having the 70 or 80% of the similarity,
ah.
so what would be the another characterization for that?
There are many multi-gene tools are available. Apart from 18S, we have mentioned that there are 4 or 5 genes are available. So multi-gene
Right.
approach, you have to go for that identifier. But the problem is
Mm-hmm.
that the sequences are not available in the gene bank,
Yeah,
identified
right.
sequence. Therefore, it becomes very difficult to get the name. We also face the similarity. We send the culture for identification of a one chitomium species of our student because of
Mm-hmm.
we were not able to identify, and what they gave us the name, which is a very common chitomium species on the basis of molecular identification. We know that the identification is not correct. Now this identification is not correct because they found that nearer what the name comes during the blasting or the phylogenetical analysis. So they put the name of that particular genre as this and mentioned
Mhm.
that this much similarity it shows. There, on the basis of this, you cannot identify if the molecular details or the sequence data, identified sequence data is not available in the gene bank, your species will
Mhm.
not able to identify it. You will have to go for culturing, morphological, and the micro characterization, microscopic characterization
Mhm.
to see the literature, and then you have to compare it with that. If the data is available, no problem. Your species can be identified very easily. So this is what the other tool. One tool is that you have to go for a multi-gene analysis, and
Mhm.
second, you have to come back to the traditional way of identification with on the basis of the available literature only.
Okay. And if I have got some special fungi,
हाँ
uh, which sequence is not going to match in the existing library,
हाँ
so उसको क्या हम नोवेल है, ये हमारा नया फॉर्म है
हाँ
फंगी का,
नहीं नहीं नहीं,
ये
अभी मैंने एक
हम
इंडेक्स फंगोरम का
कैसे
टेबल बताया था,
पूरी क्राइटेरिया,
उसमें यही
राइट?
चीज मेंशन की गई थी कि
हाँ
जो
हाँ।
उन्होंने स्पीशीज के जो आइडेंटिफाइड नेम दे ट्राई टू टेक आउट, तो दे फाउंड आउट
हम्म,
दैट देयर आर the new names have been given to the fungi which are already explained by some scientists. So this type of
राइट।
a work is going on. You have to go very comprehensively because the literature, if literature is not available,
हम्म,
then it becomes very difficult to identify the literature where it is available. You should take out the all characteristic feature, and then you can, or you can collaborate with some laboratories which are having this facility of
राइट।
literature, those who are working in the field of fungal diversity. There are many groups. The Vijayvardhane and Hyde, though they are, Hyde is one of the big name in
हम्म,
the Thailand University who is working and editor general of a Microsphere. So they help to identify such type of a fungi which are not able to
existing in
identify on the
library.
basis.
Okay, sir. Thank you, sir. Thank you very much.
Welcome. Okay,
हॅलो.
if हाँ
सर गुड आफ्टरनून.
good
मला
afternoon.
एक छोटासा प्रश्न होता, की
हाँ
ते जे टॅक्झन रँकिंगमध्ये आपण डिव्हिजनच्या ऐवजी फायलम जे लिहितो आहे आता,
हाँ हाँ हाँ
तर ते का का केलं असं? म्हणजे त्याचं रिझन
ते आता
मला
पहा,
माहिती
जे
आहे.
इंटरनॅशनल कोड ऑफ नॉमेनक्लेचर जेव्हा बनवल्या जातात, तर त्याच्यामध्ये
हं.
तुम्हाला नॉमेनक्लेचरचे प्रायोरिटी रूल्स असतात.
हो.
तर ते प्रायोरिटीचे रूल्स मग अप्लाय करताना जर एखाद गोष्ट लक्षात
हं.
आली की फायलम हा वर्ड मोर कॉम्प्रहेन्सिव्ह आहे दॅन द डिव्हिजन, मग त्याला वेटेज
हं.
दिल्या जाते. आता हे त्या रूल्स प्रमाणे त्याच्यामध्ये आता नवीन फायलम हा वर्ड यूज करा असं त्यांनी म्हटलंय. नवीन टेक्सॉनॉमिक रँकिंग त्यांनी जे ICVN डिक्लेअर केलेली आहे,
हो.
त्याच्यामध्ये त्यांनी फायलम हा, आता झुलॉजीमध्ये हा शब्दच वापरला जातो आहे त्या लेवलला.
हो, हो.
एक
म्हणजे आधी कसं असायचं ना,
इंटरडिसिप्लिनरी
की
लेवलवरती, हा, एक
हां.
इंटरडिसिप्लिनरी लेवलवरती त्याला सेम लेवलवर आणण्यासाठी तो वर्ड आता
हं.
जास्ती यूज केला. ॲनिमल साठी तो केलाच जात होता. प्लांट मध्ये जनरली
हं, हं.
आपल्या इकडे डिव्हिजन हा वापरायचे, पण ते एक युनिफॉर्मिटी यावी म्हणून ते त्याच्यामध्ये ते करण्यात आलेलं आहे बेसिकली.
म्हणजे पूर्ण प्लांटसाठीच डिव्हिजनला रिप्लेस करून आता म्हणजे
हा
सर्वांसाठी
हा
कॉमन
हा
राहील.
हा आता तुम्ही नवीन कोणतेही क्लासिफिकेशन घ्या, जे APG4 सिस्टीम ऑफ क्लासिफिकेशन ऑफ एंजिओस्पर्म आहे, तुम्हाला
हं.
त्याच्यामध्ये फायलम हाच वर्ड मिळेल. आता डिव्हिजन वगैरे नाही मिळत.
ओके, 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 ओके. ओके सर. थँक यू सर.
वेलकम.
Sir, this is Vivek Narkhedkar. I have one
Yes.
question.
Yes.
Uh, Please correct me if I am wrong because I am nothing but zero in the study of fungi. Uh, I want to know that now, as we are moving toward the genetic basis of classification, we are
Yes,
using
yes, yes.
nucleic acids.
Yes,
But
yes,
initially, we were using morphological characters,
correct.
and the deuteromycotina is based on the upset or undiscovery of a sexual stage.
Yes, yes. Perfect state
So
spore
as
is absent.
uh, as we are moving to genetic basis,
Uh,
then in the near future, will there be any relevance of that group, or do we need
no,
that group anymore?
that group is totally dismantled or depend from the new classification. You will never heard deuteromycotina word or imperfect fungi. This word has been now because now we come to know that on the basis of molecular sequencing technology that these fungi, which are we called as a imperfect fungi, when we try to find out the phylogenetic prepare phylogenetic tree, they almost come. Most of these deuteromycete fungi comes in the ascomycetes groups, and the perfect state spores are discovered by many scientists. So now this group is deuteromycotina group or deuteromycetes or fungi imperfecta. This name you will never see in new classification. This was an artificial group. We do not found the perfect state spore, therefore we prepared fungi imperfecta group. Now that group has been dismantled totally. You will never find because on the molecule the scientist said you go and discover the perfect state of that particular fungi. They are there. Some of the fungi are have lose their capacity to produce the sexual fruiting bodies, and they very well reproduce asexually. Therefore, sexual stage is not found. But when sequence similarity was found, they most of these deuteromycetes members come in the class ascomycetes. Most of them. Therefore, this group is now not relevant at all.
Okay, thank you, sir. Thank
Sir, I have one question.
Yes.
Sir, still the nomenclature of giving sexual and asexual stage, we are giving different nomenclature for the same fungi, right, sir? So
Yes.
whether it is still existing or it is also
No,
dismantled
no,
in
no,
this
no.
new system?
Yes. Now it is molecular based, sequence based. So then it is a totally paradigm change. It ranking arrangement is quite comprehensive, quite detailed on the basis of this RNA sequencing, 18S RNA or any other EPF, RPF, genes जो भी RNA polymerase वाले जो gene है जो बहुत conserve है. So this thing is now well established. There is no doubt that this methodology is now going to use, but there are some drawbacks in that also. How to delimit genera from species, species from genera from families, family from order. There is a quite discussion on scientists that this is a time, this is particular time which is a very vigorous time in the study of a fungi, and where we are not still able to comprehend or still not able to draft a rule where we should delimit this. Nowadays, the word clad is used instead of different ranking above order, above the level of orders. So, in APG system of angiosperm classification, you know that there are various clades have been mentioned because there is a problem where to delimit how much sequence similarity is present. We will call it as a family. How much sequence similarity is present, we will call it as a order. How much sequence similarity present, species. So, such type of problems are still there. So, this is one of the drawback that the perfect classification cannot be prepared on the basis of the few genes by considering few genes, but you can very clearly arrange the different genera or the species in a sequential manner. So that classification gives which one is the primitive, which one is the advanced. That ranking can be very well understand. So morphological basis and other things these are not considered nowadays as a perfect to describe any classification, because there is one more problem that have been mentioned, that the if one particular fungi found on a different host, 10 different hosts, we name them as a 10 different species. But now discussion is going on, we will not call them as a 10 different species, we will call only one fungi, one name. This is what the new uh, point which are now discussing, and the scientists are determining that each fungi should have only one legal name. So then only we will able to come to conclusion that how many species are exactly available in the nature.
Thank you, sir. Thank you.
Sir, I have one question, sir. Which
Yes.
microscope is used for good quality photograph of fungi?
Say, generally that depends, it is a quite subjective, that depends upon your budget. If you are collecting a good fungi, good number of fungi, you are very hard working, you are going to forest, collecting samples, coming to college and preparing cultures or purifying fungi, isolating them. And if you are not having a very costly microscope, if you are having a microscope just 25,000 or 30,000 rupees, there are sharp microscopes are there. They gives a very good resolutions, but they are costly. They cost near about more than 1 lakh, 2 lakh. If they are available, they are very good. And there are different staining techniques are available, and by using different staining techniques and these new microscopes, you can take a very good photograph. But if that facility is not available, if you are having binocular microscope with a trinocular microscope with camera assembly, simple camera assembly, you can carry out diversity work in any laboratory for identification.
Okay, thanks.
Any more question? Sir, I think there are no questions now.
Okay.
So thank you so much, sir. I extend my thanks to Dr. Fulzele, sir, for wonderful session. On behalf of UGC HRDC Nagpur and from RTM Nagpur University, my heartfelt thanks to Dr. Fulzele, sir, and from all the participants also, I extend my thanks to you, sir. Thank you so much, sir, for your session.
Thank you. Thanks to all organizer and participants.
Thank you, sir. Thank you.
Thank you, sir.
Thank you, all of you. Today there is only one assignment in the sense that is report submission. Today there is no ICT session recorded video because अभी आज का दिन थोड़ा अलग है, आप लोगों के घर में भी सभी पूजा वगैरह होगी, तो आज अपन थोड़ा सा ब्रेक लेते हैं, बट डोंट वेस्ट द टाइम, यू जस्ट प्रोसीड फॉर योर सेमिनार पीपीटी प्रिपरेशन. तो आज आप वो वाला टास्क कंप्लीट करिए और कल शाम तक आपके पीपीटीज भी आ जाने चाहिए. टाइटल्स तो आई होप सभी के आ चुके हैं. आई विल डिस्प्ले द टाइम टेबल बाय टुनाइट फॉर सेमिनार. मंडे से आप लोगों के सेमिनार्स रहेंगे और कल तक आप लोगों को पीपीटी सबमिट करनी है. पीपीटी आप पीपीटी फॉर्मेट में भी सबमिट कर सकते हैं या तो उसको पीडीएफ में कन्वर्ट करके भेजा तो भी चलेगा. फाइन, दोनों भी फॉर्म्स अपने जिनोमियो के साइट पे अवेलेबल है. आज का टास्क आपको सिर्फ रिपोर्ट सबमिशन है, ये 2 सेशंस का, प्लस जो सर ने वो डाउनलोड करने के लिए बोला है कल के लिए जो रेडी रखना है वो भी आप थोड़ा सा देख लीजिए अगर पॉसिबल है तो.
Thank you.
Okay, ma'am. Thank you.
Okay, ma'am.
Okay, ma'am.
Thank you, ma'am.
Thank you, ma'am.
ओके, थैंक यू ऑल ऑफ यू. कल मिलते हैं सुबह 7:15 बजे. थैंक यू
Yeah.
ऑल ऑफ यू.